Today, I'm talking about statistical guidelines on sex inclusion in clinical studies at the National Institutes of Health. I did this work while I was employed by the NIH, and currently I'm with Regeneron Medicine as a biostatistician. So, the outline for my talk is an introduction of how this topic came about and introduce the 4Cs framework that NIH has come up with, and then leave you with some final thoughts. So, the beginning of this work is the 21st Century's Cures Act or mandate. It's a congressional mandate that was signed into law actually by the president, the former presidents, and it passed by Congress in 2016 and was designed to inject innovation, accountability into biomedical research and speed new treatments to patients. It's a huge mandate, and it funded major initiatives like the Cancer Moonshot and the Brain Initiative and Precision Medicine, etc. And while doing so, the Congress also mandated reforms at NIH. One key reform was the requirement that sex as a biological variable, SABV, be integrated into all NIH-funded research to improve reproducibility and generalizability. I worked at the NIH Clinical Center, and there are 27 institutes at NIH. Clinical Center is the research hospital. So I was asked to turn this policy mandate into practical statistical guidance. In other words, the act set the vision, and my work provided the framework for how SABV could be implemented in study design and analyses. The 4Cs of studying sex to strengthen science is consider, collect, characterize, and communicate. Consider design studies that take sex into account and explain or explain why it isn't incorporated. For example, you could be studying prostate cancer or breast cancer. Collect the data. So in other words, you have to have the instruments to collect sex information, characterize, analyze based on sex, publish your results, and disaggregate based on sex. So as we know, there are observed differences between men and women. I think everybody can talk about that in many different therapeutic areas. Cardiovascular is one. Recently, we heard about beta blockers not being that effective in preventing stroke in women. Osteoporosis, heavily studied in women. That means the men are underdiagnosed and undertreated. These are just some examples in the media. But there are also many studies that show there are different reactions to drugs. There have been studies that show different manifestation and intensity of pain, and obviously incidence of diseases and conditions. So this 4Cs framework, it was invented by ORWH, that's Office of Research on Women's Health at the NIH. So what I did was adopt this framework, stayed true to this framework, and then framed the statistics around this framework. So I start with a motivating example. It's a simulation. It's a simulated data set of heart attack. Let's start with the 90 subjects. 2/3 are males and 1/3 of females, not unlike what was happening in 60s and 1970s and later. Let's say 70% of these subjects had heart attacks. Of those with heart attacks, males had chest pain with probability 80%, and females with probability 30%. Males had nausea with probability 25%, females with 80%. Males had fatigue with probability 25%. females had fatigue with probability 65%. And of those without heart attack, risk of symptoms were just 10%. So the first model is model one. You model the probability or the logit of the probability of heart attack with chest pain. And from logistic regression in my simulated data set, you would obtain chest pain, obviously, because I set up the data with very high significant threshold here, with an odds ratio suggesting a strong association between chest pain and heart attack. The intercept is not just significant, indicating the baseline odds are close to neutral. Now, adding sex yields a statistically significant p-value, marginal. I think this is an extra zero. Sorry about that. So that could be a clue that something is happening if you added sex here. So the interpretation is that this model captures the historical bias, for example, associating chest pain with heart attack. Basically, this is reflecting the male majority data set. So chest pain alone is a strong predictor, but it ignores potential sex-based differences or additional symptoms. Adding sex revealed a potential baseline difference between men and women in heart attack risk. But the marginal significance of sex, which is p-value 0.05, shows that chest pain may not fully explain sex-based differences, indicating the need for additional variables. So here, sex is a proxy variable, actually, for unmeasured female-specific symptoms for heart attack, showing that SABV would have mattered had we done this 30 years ago. In the second scenario, I increased the sample size to 210. So now I have nausea in the model. So I've done a couple of things. I've increased the sample size, so there's more females, and I've added a symptom. Chest pain remains highly significant. Nausea becomes also significant. So that shows right there, it's something called omitted variable bias, right? So you can only have an association based on the model you have, but you cannot really generalize the whole business of what predicts heart attack. So sex, when I do this, sex is no longer significant, suggesting no difference in the baseline risk of heart attack between men and women. But the interaction that we know the interaction exists because we simulated the data is not explained by this model. And it doesn't guarantee that the omitted variable bias is gone. So nausea, a symptom more common in women, showed up as significant. First, because it's in the model, and secondly, now the sample size has been increased. So the effect of sex is lessened, indicating sex-specific differences. Sorry, I have to do this here. Sex-specific differences is lessened. So adding the nausea, what happened is adding nausea disentangles its effects from those of sex. So allowing the model to properly adjust for sex differences. So this reveals the true baseline effect of sex, but it doesn't show the interplay between sex and symptoms like nausea. Model 3, I bring in fatigue, and fatigue now, chest pain is significant, nausea, adding nausea, obviously reducing omitted variable bias, fatigue also is significant, and sex remains not significant. But still, the interaction effect is not explained because no interaction terms are in the model. So if you just stop there, then you would say, well, you have a pretty good correlation model. So including nausea and fatigue, symptoms more common in women, highlighted their importance, but the effect of sex is not significant. You could report that, that would be honest. But sex-specific differences, whether they are differences for each symptom, is not captured by this model. And model 4, so now I've increased the sample size, has the main effects, sex, and all the two-way interactions with sex. So now I have fatigue, nausea, and chest pain significant. Sex is not significant, but the interaction terms are significant. So the loss of significance of sex suggests its effect is now fully absorbed or explained by the interaction terms. And chest pain remains significant for men via the interaction, and nausea and fatigue emerge as critical predictors for women. So it demonstrates sort of this toy example, the complex interplay of symptoms and potentially management in the emergency room by sex in this specific simulation case. So at the evolution of knowledge is said that n equals 90, chest pain was associated with the probability of heart attack on mostly male data set. Incorporating sex would have given you a clue, not the entire story, but it would have been a proxy variable. Then we increased the sample size, we got recruited new patients, we added more variables, and we found other symptoms were important. And finally, in n equals 450, sample size was increased and interactions were found showing the interplay between sex and symptoms. So that's to motivate the talk and to tell you that I came up with the guidance so that it's first of all clear to the PIs what to be very, to actually set some framework for them from the very beginning, and also when to request statistical help from statisticians. So one of the main thing is inclusion of sex in the protocol. You have to collect sex as a variable, first of all. Believe it or not, I've seen studies where it was not collected. So, and then also decide how sex will be analyzed in the study, descriptive, exploratory, confirmatory. Ensure that there's recruitment strategies for your disease-specific case. If a cardiovascular, you want to have a good representation of both sexes. You can use stratified sampling or oversampling later. If you can oversample if one sex is underrepresented. But if you don't have a strategy, then you are, left with, for example, convenience sampling in an observational study. Single studies are okay, single sex studies, as long as you have a justification for doing it that way. So recruitment strategies aims for a balanced recruitment, and it should reflect your study population, essentially. If there is more than one sex that's included, then we have to then decide whether it's exploratory, where you're going to confirm sex as a variable that you're going to do inference on, or it's going to be a exploratory variable post hoc analyses. So if sex is an exploratory variable, then we have to decide how to operationalize it. For NIH Clinical Center, design clinical trial or observational studies, select recruitment strategies to get an ideal mix, to mitigate external validity biases, decide on reporting strategies, explore how you're going to collect the data, and are there other confounders of sex? For example, if you knew, and this happens that a particular genotype is more prevalent in female, then you know that that is a confounder in which you also need to collect that information. Otherwise, you would be reporting information by sex when it's really something else. Determine whether you need to stratify. Are you going to randomize by sex as a stratification factor just for randomization? Are you also going to adjust for it in the model? You may not, and most often you will not have power for statistical inference by subgroup if you didn't plan for it. So it is important, if it is important that you explore your outcomes by subgroup and do actual p-value reporting, then one should contact statistician. For clinical trials, sex is often used as a sensitivity analysis. So this is quite different. This is not a setting where you're trying to find differences by sex. You're almost trying to show that your treatment effect is robust after you adjust for certain demographic factors like sex. And if it's not robust, if it's changing your treatment effect, then you still cannot say that's a confirmatory finding. It is still exploratory. A sensitivity analysis with regards to sex is an exploratory result in a trial protocol. If you're confirming a difference by sex, again, you have to decide how to operationalize the collection of the sex. Stage one would be you want to test for and confirm a difference by sex. You should power for sex. So you may need to stratify. You may need to use a full model with your treatment effect and an interaction effect or disease outcome effect and an interaction effect. Stage two is you may want to report results separately by sex. And in that, you have to do formal subgroup analysis. Here is a guidance. Here's a link by Estelle Rusek-Cohen and Richard Simon of FDA and NCI that goes into evaluating treatments when a gender by treatment interaction may exist. So this is a great primer for subgroup analyses. Now, I've been talking about, now we're in the second C, collect. How do we collect it? So what I suggested for the NIH is to have sex at birth for sure, female and male, and also to include intersex. I also have sex at present, and I'll get into that in a bit. So it's usually self-reported. Sex is usually self-reported, but you can also have physician-reported if it's a pediatric study, for example, or a caregiver. And so here is another ways. Highly recommended is sex at birth. If you have sex at birth, and then you can have, if you have current sex, I can actually derive the variable of cis male, cis female, trans male, and trans female. And intersex is also quite important. Many pediatric studies and many other therapeutic indications, it's important to collect that. And then, so I talked about self-reported, but of course, you can also do a genetic test. And if you do, then you must have XX, XY, and other. So now let's talk about sex as exploratory, which is 90% of where people are. And so the first way to look at it, the most common, is to demographic. Who was in your study? So you can do, you should report sex by race. It's an FDA mandate, actually. Then you can do descriptive study. And up to here is what NIH mandated. You must disaggregate your study finding. Could be a treatment effect, for example, by sex. You don't have to give any inference, any p-values, but just tabulate it. Even if not focused on eliciting SABV difference, it's important to explore relevant outcome, biomarker, or imaging data by sex and gender and other groups. For example, so here I'm mostly talking about descriptive statistics. When you want to do inference, then you should consult a statistician. So so one thing, as I mentioned, there's the sensitivity analyses. It's usually done in the setting of clinical trials. The interaction analysis is important, and you should pre-specify this in your statistical analysis plan. You can also do subgroup analyses where you can get a within sex estimate of treatment effect. You can do a stratified test or a model-based combined. After doing this stratification, you can do a combination of the strata for everybody, the treatment effect. You can also enable an A versus A complement, so male versus female. And if it's insufficient, for example, if you have mostly male, then you can probably give a treatment effect for A and all, meaning males and all patients but not for females, for example. So these are three of the most common ways of doing inference. One is sensitivity, interaction, subgroup analysis. Now, there are other complex analyses. You could do data augmentation if you have sex imbalance, for example, using propensity score matching. For reporting, many people use forest plots, and so you can look at treatment for male, female, or intersex, and other demographic variables. But it gets a little bit tricky, I would say. I would say that do not include p-values. Actually, New England Journal of Medicine does not want, for example, now p-values with forest plots because these are unplanned, right? So these are still considered descriptive ways of, if you plan for it, then go ahead. If you plan and doing an interaction test and empowered for it, then you can do it. There are other ways. You can, of course, do a a lot of people say they have done the Bonferroni correction. Usually, in my experience, that doesn't really work because you've tried like 300 hypotheses and you've got 10 in a forest plot in your paper. So it's not really justified to divide p-values by 10 in that case. So maybe it is better to do maybe some Bayesian shrinkage method if you are interested in actually giving some effect size, corrected effect sizes, and p-value or p-value like probabilities. So now I want to go, so now we're going to characterize in all analyses above, study is most likely going to be underpowered to detect differences and make a real inference on sex. But you can generate effect sizes, sort of hypothesis generating, if you will, if you see a large difference by sex, to identify signals to confirm in future research. So I would say when you characterize it in scientific literature, do not point to it as evidence or definitive. There may very well be other confounders if you even have found something for sex. And conversely, if you didn't find something, doesn't mean that actually it's good if you didn't find something, but that doesn't mean necessarily. Again, I showed you through the example that you may not have unveiled all the interplay with sex in variables. So that's why when you characterize your findings, you have to explain the strength and weakness of your study and of your analysis. And that is true of any good science. So it isn't about finding a p-value. It is about, does it make sense biologically? And what are the weaknesses of what I'm reporting and of my experiment? So again, I recommend for all principal investigators to consult statisticians from design phase onwards within sex analyses of study outcomes. If it's desired but not powered, please consult a statistician. Another case is investigator found sex differences, which was not expected. Okay, but before you write it in a paper, we need to review this. If you had a small study, if you had a rare outcome study, there may be a lot of challenges present in your recruitment strategy for having a sex ratio that is relevant in your disease area. In all of those cases, you must consult a statistician. In many ways, I put this priority chart together. Start with descriptive, then you go to sensitivity, interaction, multivariate modeling, and then other integration of other external data sets. You can see that the importance of consulting a statistician goes up significantly with ChatGPT and other tools. A lot of investigators are comfortable doing this, and that is fine. But as you get more and more nuanced in your trying to unlock differences by sex, you will need the help of consulting statisticians. When you're doing a confirmatory SABV work, you have to power your study. Definitely, you need statistician to power your study. You have to consider the interaction effect. You have to consider stratification, potential confounders, and carefully interpret clinical and biological importance of effect size and importance by sex. Communication is that researchers should report if study design incorporated sex-inclusive practices, like we went through, and the do's are do report a biological finding by sex, explain differences you found, especially clinically, not on a p-value basis, consult statistician for appropriate design, sample size, and include limitations and strengths of in your paper. What you shouldn't do is, if exploratory analyses with respect to sex don't emphasize a p-value only or a lack thereof, so that there is no difference. And I think that, this is more about communication, efficacy outcome, or other variables. You can include bisex statistical summary, the EDA, the exploratory data analyses, forest plots, as I mentioned. I do not recommend including p-values in those. Safety and PK/PD, it's very important actually to disaggregate by sex, not just your efficacy endpoint. Do not stress causality. That is one of the big pitfalls. Exercise caution when interpreting statistically significant result. More than not, it is a spurious finding. You can include sensitivity analyses. You can include interaction analyses. You can include multivariate analyses and data augmentation methods and many other methods, but you will need the sound statistical underpinning in your paper. So I think that the communication there has to have a clarity in reporting. You have to have the contextual interpretation. Why is this important in where you're studying? Communicate limitations, and the visualization of results is going to be very important as well. So I think I will leave you with these two references and just open it up for questions, comments.